So good day, everyone. Again, we will have another topic, and this will be the last part of our course subject, which is all about fungi. And this is the mycology part. Mycology, of course, deals with the study of fungi, which includes multicellular and unicellular fungi, the molds and yeast. So we will discuss what are the general features of fungi associated with disease in animals. So there are approximately 1.5 million species in the kingdom fungi. Before it was regarded as kingdom. But of course, this fungi was under the domain Eukarya, which includes also the plant cell, animal cell, protist, and also this fungi. Approximately, there are 100,000 species described, and 400 of these fungal species are known to be pathogenic in man and animals. So there are 5 recognized phyla. These are Microspora, Ascomycota, Basidiomycota, Blastocladiomycota, and Glomeromycota. So for the general characteristics of fungi, they are eukaryotic, non-photosynthetic microorganisms widely distributed in the environment. The cell wall contains chitin and other polysaccharide. It has branching hyphae and unicellular yeast are the 2 main morphological forms. So we can see here that the septate branching hyphae of a mold. When you say septate, this is divided by these cross walls. And a mass of interfacing hyphae forms a mycelium. And these are budding cells of a yeast. So the characteristics are they are known to reproduce both sexually and asexually with the production of spores, but the spores are different from the bacterial spores produced by the genera Bacillus and Clostridium. Most grow aerobically at 25 degrees Celsius. Some yeast are facultative anaerobes, and some ruminal fungi are strict anaerobes that are present among ruminants, rumen. Tolerate both high osmotic pressures and low pH values, resistant to antimicrobial drugs, which are effective against bacteria because specific chemotherapy used for fungi are antifungal drugs. Majority are saprous or saprophytes. Some cause opportunistic infection. Some fungi produce toxic substances, which are known to be mycotoxins, which are secondary metabolites produced by fungi, causing disease both in humans and animals. So for these fungi, we have different groups that we will discuss. The dermatophytes that are known to cause ringworm in animals and humans. We have opportunistic fungi, which are the Aspergillus species, and they are known to cause opportunistic infections, and they are also known to produce mycotoxins, the aflatoxins that can affect both human and animals. And then we will discuss also the yeast that occur as commensals on the skin and mucous membrane, causing opportunistic infection. We have also the dimorphic fungi that occur as mold in the environment and yeast form in animal tissues, also known to cause or produce opportunistic infections, both animals and humans. Then we have the zygomycetes that are known to be widely distributed saprophytes or saprobes, causing sporadic opportunistic infection. And the mycotoxin producing species that are known to produce these mycotoxins that are the toxic secondary toxic metabolites growing in crops, stored feed, or pasture litter. So the three phyla distinguished by their sexual forms are Ascomycota, the Ascomycetes, Basidium mycota, and Zygomycota. So the spores, sexual spores now are ascospores for Ascomycota, basidiospores for Basidium mycota, and zygospores for Zygomycota. So for the characteristics that we discussed a while ago, so they are eukaryotic, non-photosynthetic. They can be macro or microscopic, widely distributed in the environment. The cell wall contains chitin, which is the main component of the cell wall of fungi, and other polysaccharides. They are heterotrophic organisms known to produce exoenzymes and obtain nutrients by absorption. There are unicellular, which are the yeast, and the multicellular, which produce branching hyphae, which are known to be the molds. And this hyphae is the tubular structure like of these molds. Mostly they inhabit terrestrial habitats or land, but some were adapted to aquatic life. They are aerobic at 25, especially at 25 degrees Celsius. Some molds also are known to be strict aerobes. So they reproduce both sexually and asexually with production of spores, tolerate high osmotic pressure and low pH values, grow on SDA, pH 5.5, resistant to antimicrobial drugs. Majority are saprophytes. They prefer cool and damp niches. These are the structure of a typical eukaryotic cell. So since they are eukaryotic cell, they have structures like the nucleus, the nuclear membrane, and other organelles. So the hypal cell wall is composed of carbohydrate, which is a chitin, macromolecules with cellulose cross-linkages that imparts rigidity and osmotic stability. The presence of ergosterol in the cell membrane is known to be the predominant sterol. They have nuclei and well-defined nuclear membrane and other membrane-bound organelle like mitochondria, with the presence of network of microtubules, and the septa, which is the presence of the cross wall, is often present in hyphae. That's why these hyphae are being called septated hyphae. There are also hyphae without septum or the cross walls, so they are known to be synocytic hyphae. So dimorphism is also characteristic of some fungi organism, wherein the fungus can appear or can be present in two forms depending on the conditions. So when it is subjected to 37 degrees Celsius, it can be yeast, or in the animal tissue, it can be yeast. When subjected to lower temperature like 25 degrees Celsius, it can be mold, which is known to be the filamentous fungi. And they could also be opportunistic pathogen. So these are the sexual spores that we mentioned a while ago, produced by the three phyla Ascomycota, producing ascospores, Basidiomycota producing basidiospores, and the Zygomycota producing zygospores. These are the three sexual spores of fungi. So the incubation conditions appropriate for the aerobic culture of fungi, based on what on the groupings of this fungal organism. For the dermatophytes, they are usually cultured at 25 degrees Celsius for 2 to 4 weeks. Aspergillus species 35 to 37 for 1 to 4 days. Yeast, the pathogenic yeast are known to grow well at 37 degrees Celsius for 1 to 4 days. And then we mentioned the dimorphic fungi. When they are mold, they are cultured at temperature, or they are growing at 25 degrees Celsius for 1 to 4 weeks. Yeast phase, 37 degrees Celsius for 1 to 4 weeks. And we have also the zygomycetes growing at 34 degrees Celsius for 1 to 4 days. So these are the unicellular fungi, non-filamentous because the filamentous are the molds. They are spherical or round or oval shape. They are with membrane-bound nucleus, and some are facultative and aerobes. The filamentous fungi, these are the multicellular organism. Long branch filament is called hyphae. And an aggregation of hyphae or mass of hyphae is known to be mycelium. And for plural, it is mycelia. They are aerobic fungi. So we have the suborod dextrous agar, or the suborod agar, named after a French dermatologist, Dr. Raymond Suborod, who is a specialist in scalp disease, invented or used a selective media containing sugars and peptone and has a low pH, which is inhibitory for most other microorganisms, but for fungi, they are able to grow on this low pH. So it was named after Dr. Sabarod, the most commonly used culture media for cultivating fungi, which is the Sabarod dextrose agar. For the growth, reproduction, and colonial formation, so the airborne fungal spores are able to germinate in locations where environmental conditions are favorable. Spores swell and their metabolic activity increases prior to production of tubular projection, which develop into branch hyphae. Molds tend to form large colonies, which grow with growth and extension of hyphae at their peripheries. So for the reproduction, as we mentioned, they can undergo sexual or asexual form of reproduction. For the asexual form, can be through budding, where the parent cell divide into equal or unequal daughter cells, or through production of spores. Example of the asexual spores are sporangiospores and conidiospores. So these are the different asexual spores produced by fungi of veterinary importance. So we have the arthroconidia or the arthrospores. They are formed and subsequently released during the process of hypofermentation. This is characteristic of Coccidioides immitis and also by dermatophytes, fungal organism. Blastoconidia or blastospores is produced by budding. Example here is the Candida albicans that is known to produce blastoconidia. And then we have also the chlamydia conidia or the chlamydiospores, thick-walled resistant spore which contains storage products. Macroconidia, they are large multi-celled conidia produced by dermatophytes. Also, the microconidia are produced by dermato, but micro means small conidia. Phyaloconidia, they are produced from phyalides, and sporangiospores formed by zygomycetes, released when the sporangium ruptures. An example of the fungi that produce sporangiospores are rhizopus. So these are the different asexual spores produced by the different fungi organisms. Next, we have the mechanisms involved in fungal disease. We have the tissue invasion, which is, we call it as mycosis. The general term for the disease caused by fungi is mycosis. And when the Fungal disease is caused primarily by the toxins produced by the fungal organism. We call it mycotoxicosis. And they can also cause disease by induction of hypersensitivity. So the general features of fungal disease, they can cause superficial mycosis. It involves mainly the epidermis and other superficial structure of the skin. And most commonly associated with this are the dermatophytes that causes dermatophytosis. And then there are also some fungal organisms that are known to cause cutaneous mycosis. We have the Candida and Malassezia pachydermatis. So we categorize fungal diseases according to their sites of lesions. We have superficial mycosis when it affects the epidermis and other keratinized structures and mucous membrane. Subcutaneous mycosis when it affects subcutaneous tissues. Systemic mycosis when it affects reproductive, respiratory, digestive tract, and other organ system. So it is generalized or systemic. There are factors that may predispose to fungal invasion of tissues like immunosuppression, prolonged antibiotic therapy, immunological defects, immaturity, aging, malnutrition, and exposure to heavy challenge of fungal spores, traumatized tissues, and persistent moisture on the skin surface, like in the cases of ringworm infection, and the presence of some neoplastic conditions. So for diagnosis of fungal diseases, we can base our diagnosis based on the clinical signs and history, especially for dermatophytosis. We can submit specimens for diagnosis by submitting hair and skin scrapings, especially from superficial mycosis, biopsy or postmortem specimen from subcutaneous and systemic mycosis, and direct microscopic examination of wet preparations may be confirmatory. Culturing in Subrhodexus agar and histopathological demonstration of fungal hyphae or yeast forms. How do we differentiate fungal species? By their form of the sexual form or the telomorph. It is used for assigning a fungus to a phylum. Examination of the sporing head for conidial arrangement, type and morphology of spores, the features of vegetative hyphae, the presence or absence of septa or the cross wall, by their production of pigment. If it's pigmented, we call it demachaceous, and when it is colorless or hyaline. Specific hyphal structures such as rocket shape and spiral shape. By their colonial characteristics, size and appearance after specified incubation time, color of both the obverse and reverse side, surface elevations or depression, colonial appearance, size and shape of individual cell, biochemical reaction, immunological tests of dimorphic fungi, by their soluble antigens that they can produce, and the specific nucleic acid probes for dimorphic fungi. Then we have also the antifungal chemotherapy. We have polyene antifungal drugs. They are known to bind selectively to ergosterol, which is the predominant sterol in the cell membrane of fungi. Nystatin and amphotericin B are example of these drugs. And then there are also antifungal drugs known to inhibit the synthesis of ergosterol, and these are the antifungal azole, like ketoconazole, itraconazole, voriconazole, itraconazole, and then we have also antifungal drug used for the treatment of ringworm, like griseofulvin. So this is an image showing the production of yeast through budding. We have here the budding yeast produced by the Saccharomyces. For example, Saccharomyces cerevisiae. And this one is the yeast undergoing fission. So these are some conidiospore, which is not enclosed within a sac, while the sporangiospore is enclosed in a sac-like called sporangium. So these are some of the pictures of mold growth in a plate. And this one is an opportunistic mycosis that causes opportunistic mycosis like the aspergillus, but this one is Aspergillus niger with this black appearance. So this ends the introduction of the general features of fungi associated with disease in animals. Thank you.